when I got the invitation from Ron, it was a little bit shocking because I think I'm about 10 years too young for this particular type of talk. I wasn't really ready for this sort of retrospective view on how I got where I am and even thinking about how to communicate it to others so they might learn something from it. But it's been a learning process, and it's been extremely interesting being here, hearing the other other senior scientists reflect on their natural history, their career choices, and how they got where they were, and communicate what they think is important for young people. I've been really inspired by the talks I've heard, and what I want to do is build on a few of them, a few of the major themes that I've heard, and tell you a little bit about some of the things that have been important for me. First, I need to recognize that I'm sponsored by the EMBO, and as you heard from Anne-Marie, India is now part of the European Union as far as science is concerned. And you should take advantage of that, because EMBO is a really wonderful organization. It just emphasizes absolutely top quality science, and their fellowships are worth their weight in gold. So really take advantage of this change in things. I'm hoping that I'll be able to highlight some of the research done by 5 really outstanding scientists, Indian scientists who I've been able to work with over the years. They're listed here. And I think what I want to do is to jump into 4 themes, really to build on what Ari Ari's 13 themes that he had brought up, but I have sort of 4 that I'd like to emphasize, and that resonates with some of the other ones. First of all, a scientist is a knowledge addict. And like all addictions, whether or not it's the good things like, chocolate or cricket, or things that you might want to not be too addicted to, like various types of drugs and sex and whatever other things. But regardless of what you're addicted to, it involves a dopamine circuit. I think science is really no different from any other type of addiction that we have. It's an addiction to discovering something new, to getting that, that dopamine surge that you have discovered something new, those bumps in the back of your neck that rise, and so forth. And I think so much of being a successful scientist is maintaining that addiction, nurturing that addiction, and developing the ability to be addicted in the long run. And that takes a number of strategic planning steps that I'm going to argue throughout this talk. The first thing that you should do then really is to make sure that you share that addiction with your students, and that you train them to become addicted too, so that you do as much as you can to convey the science, the excitement and passion of that discovery process. And the other thing is, as we talked about earlier, the importance of selecting students in the very early stages of your career. One bad student can really kill your lab for a long period of time. And it's pretty easy to phenotype people who are not really addicted to the knowledge discovery process. They wander, their eyes think more about something else, their career, whatever else. Don't work with them. Don't select those types of students for your lab. And then I think a little more difficult in the advice side is that make sure that your partner understands this addiction. And a number of other speakers, I think have articulated exactly the same argument. I'm just trying to make sure that I get it right. So Ari, for example, mentioned that he was a little nervous about talking about his love for science, about his wife, because his wife might feel jealous about it. Anurag had called it polyamour, I think was the term that he had chosen. Not polygamy, but money loves, polyamour, right? And Fred, I think, articulated it beautifully, saying that you should do what you love and love what you do. And the way I articulate it to my students is that I do it in terms of a little bit of a ribald joke that perhaps works best for Western audiences than it does for Indian audiences, but I'll try it anyway. It's a little bit non-PC. I know it might go against the rules of the yin, but it's about 3 professional women, and they're basically discussing amongst themselves the pluses and minuses of having so we say extracurricular affairs, or whether or not one should solidify your relationship in marriage, or whether or not you should keep it loosey-goosey and just keep it at a boyfriend level. So it's a doctor, a lawyer, and a scientist. And the doctor says, all these sexually transmitted diseases, marriage is a really good thing. Keep the monogamous thing going so that you have a minimum chance of getting some of those nasty diseases. And the lawyer, of course, argues 50% of these relationships don't work out. Legalizing it, codifying it, marriage only makes it more complicated to change it. So keep it loosey-goosey and stay with the boyfriends. The scientist, well, she was looking at both the lawyer and the doctor and just sort of shaking her head and saying, you have to have both. A husband and a boyfriend. Why? Well, the reason is that you can tell the hubby, I gotta spend some time with the boyfriend, And then you can tell the boyfriend, say, I gotta spend some time with the hubby. And then you can go off and do an experiment. And I think that's really the nature of being a scientist, is that you have another love. It's a polygamous relationship. And if your partner doesn't understand that you're involved in a threesome, then it's going to be a problem. And, I lost a 30-year marriage because I didn't manage that right. And I just, think about that carefully. And I think one of the things that the best pieces of advice I could give to young people is don't let that relationship, that passion with your science become divisive. Incorporate your partner into it, incorporate your family into it, make it a family thing that brings you together rather than, fights for your attention and look at it sort of you spend so much time in the lab and so much time with the family. So I think that's a heartfelt piece of advice I could give. The second piece of advice I want to give is, there's only 4, don't worry, is the business about what actually lasts during a scientific training program. Methods come, methods go, it's the thinking process that's the lasting part. In my laboratory, there's a whiteboard in front of everybody's desks, and everybody who has a desk has to have a hypothesis and how they're going to falsify that hypothesis on their whiteboard. I really think that this is something like scientific oral hygiene, something you've got to brush every day and clarify your hypotheses, refine them, make sure that you've got good tests that are actually trying to falsify that hypothesis, and that you are continually stimulating that dopamine circuit that helps you really feel excited when you've done it. And the other side of it is that, particularly for those who are venturing out of purely mechanistic research and going on to research that asks questions about function, about evolutionary origin, or even development, that you have to be very careful that your hypothesis is posed at the right level of analysis. You can pose hypotheses at different levels of analysis, and if you're setting alternative hypotheses at different levels of analysis, you're not going to make progress. And that's something that people who, if you're not experienced and not really constantly thinking about it, can get themselves into a quagmire where you're not really posing alternative hypotheses against each other. So that's an important thing. And then, of course, just the glorious thing about having alternative hypotheses. And, this is one of the greatest gifts of being a scientist, is that you don't need to tie up your ego with one particular outcome. You've got an alternative that you can go to. So you're not, it's not a devastating thing that when you have falsified and shown that your favorite pet hypothesis is wrong. Well, great, you've got another one, right? That's the whole nature of the scientific process. And it's not really that I started thinking about my parents in this context that I realized how special science is in that regard. And of course, just as an addition thing, that when you have alternative hypotheses, you can design experiments where all outcomes are interesting. Try not to design experiments where you only have one particular outcome that's interesting and publishable. Otherwise, your productivity is going to be lower. You're going to have, you're going to be confronted with the depression that comes with falsification of your favorite hypothesis. It should be a rejuvenating process. But anyway, I want to talk about parents, and maybe a little bit about birth order too. Indians are so much better about recognizing the influence of parents at various stages in their career, and I think Western folks are not. And I want to talk a little bit about mine. So I'm number 2 out of a family of 4, and both my parents, depicted here, are medieval historians. Historians do not have alternative hypotheses. They have a single hypothesis they work on until they die. And then the field changes when a new person comes in to work on it. And I can't tell you how liberating it is when I think about how easy it is for me as a scientist to do my work compared to what my parents have dealt with. A lot of times you respond to your parents' upbringing in both positive and negative ways. My parents were just incredibly devoted, maniacal historians. My older brother became a historian. I was number 2, so I didn't have to deal with that. And birth order is really important. One of the shaping things in my life was realizing just how crazy academic life was and how little my parents, because they were such good medievalists, understood about current modern day life, you know, how things worked, what people did for a living and so forth. So I became a mechanic very early on in my life because I wanted to know how things work, and I wanted to sort of do what other things that other people had done. And so I really became a scientist as a mechanic. That's how I learned to test hypotheses. That's where I learned the pleasure of getting hypotheses right and having alternatives. Because when a car rolled in those days, you didn't have a piece of electronic ignition to plug it in and tell you what the problem was. You had to phenotype the whole car to figure out what was wrong. You know, the car would putter in, it would have these certain behaviors, and you had to figure out whether it's a carburation or electronic problem or something with the transmission or whatever. And sometimes, of course, you had to phenotype the drivers of those cars because, you know, the driver was part of that car's ecosystem and its problems. And in those days, fuel pumps didn't differentiate between diesel and gasoline, and an awful lot of drivers, you know, would fill up with the cheapest, which wasn't appropriate for their car. And that's how I met my wife. She was reading Chaucer and filling up with the cheapest, filling her Toyota Corolla, a 1967 Corolla, which was definitely a gasoline engine with diesel. So was where the beauty of learning how to falsify hypotheses, because the customer would be happy and you would be happy, and you had a really clear outcome on that process. Now, my wife was a poet. She was reading Chaucer, and she taught me that scientific English is not the same thing as spoken English, and that scientific English really is a foreign language to everyone. And what I think one of the biggest mistakes that editors frequently say is, this manuscript needs to be, you know, seen by a native speaker. Native speakers are frequently the worst writers of scientific English. Why? Because they don't think of it as a foreign language, and they don't study it as a foreign language. And some of the worst writers in my group are Americans who, you know, think that they can just write as if they were speaking. And that sort of sloppy, colloquial approach that I, as an American, have, you know, in my approach toward speaking simply doesn't work in the written word. And some of the best scientific writers are ones who have really had to learn English as a foreign language because they've learned scientific English as a foreign language. And it is something that one needs to work on continuously, because the ability to communicate well in your written will is going to determine your impact as a scientist in the end. If you cannot write well, if you do not express yourself clearly, it's going to greatly impede your ability to have a scientific impact. So it is something that you should work on and take seriously, and certainly just because you're a native speaker, you know, don't think that it's something that you shouldn't do. The second really is write while you experiment. Those two are completely integrated processes. If you're not writing while you're experimenting, you're not really confronting your data and thinking about the relationships between results and conclusions and where your path is going. It is just a process that you have to integrate together, and you certainly shouldn't allow your students to put it off to the end. It's something that if you do in a small dose spaces, one paragraph at a time, you know, papers just grow on their own and becomes a pleasurable, dopamine rewarded process, rather than something painful that you have to confront at the end when you do have to write up and show that you've done something. And this is a point that I want to bring up because it really relates to what Ron was talking about in his talk, namely that the bar has been rising higher and higher, and that in order to get a high impact publication now, you can no longer do it as a single author, single PhD project type of source. It takes 10 PhD projects to be a really high impact paper these days. And here is really a moral dilemma for a young group leader. The moral dilemma is what's best for you, of course, is getting those high impact papers and getting 10 students on a paper. And that means that each one of those students gets to write one little paragraph, the methods portion for that, whatever they contributed to it. They don't get the chance to write the big paper from start to finish. They don't have that very primal experience of writing a paper from scratch. And if you don't insist that your student does at least one of those in their PhD career, you are not training them well. Because when they go off on as a postdoc and they've got to do that on their own, and they're the one who has to actually write the first draft of the paper, it's going to be terrifying. So no matter how low of an impact the paper that is that they're going to produce, insist that they do it. There are many venues, there are many wonderful, even protocol type of papers that allow people to experience that process of having sat down, confronted a blank piece of paper, constructed an entire scientific paper on their own, and they know they'll be able to do it again. So that's what I would. Now I'll talk a little bit about some teachers. I went to Dartmouth College, was trained as a chemist and as a biologist. It was really Jack Schultz who I met there, who got me sort of corralled, I think is the right word, tamed in such a way that I became a real reasonable scientist. I ran his lab for a couple of years. And then I had the extreme fortune of being able to go get my PhD with Tom Eisner and Jerry Meinwald, who were really the fathers of chemical ecology at Cornell University. And Tom Eisner really gave, I guess May Berenbaum, who's one of Tom Eisner's students, who's sandwiched here between me and Tom, articulated it beautifully. May's a movie buff, and she collects movies about insects and sci-fi and things like that. So she thinks a lot about sort of cartoon images and the like. And lots of cartoon images have people with special enhanced features. So like Superman, he had X-ray vision. He could see through walls and whatever it was. Well, Tom Eisner had a special type of superpower, which May called nature vision. And that was the ability to be able to perceive things in a natural setting that really crystallized out a question like very few other people. And this was a nucleating moment for me, a nucleating moment that influenced how this institute was developed in 1996 that Ron mentioned, and how a school that we built at the institute was developed. So the institute basically brings together a number of different disciplines to create chemical ecological research. So chemical ecology are the ecologic interactions that are mediated by chemical signals. And to build this institute, I brought together the most ecologically oriented organic chemist, Wilhelm Baldwin, and the most ecologically oriented enzymologist of secondary metabolism, Jonathan Gershenson, and the most ecologically oriented population geneticist, Tom Mitchell-Olds. And we were the 4 founding directors of this institute in Jena. And we've since brought on a neurobiologist, Bill Hansen, and Tom Mitchell-Olds went back to Duke, and we were joined by David Heckel as the entomologist later on. So we have 5 departments, but I want to talk a little bit about the school. And the school, well, let me say a few things about Max Planck Institutes. Max Planck Institutes are unique in that they provide long-term patient funding for directors. We get lifetime support. And that means that back in 1996, I had 25 years worth of funding to think about what to do. What was going to be the vision, what was going to be the big thing to do. And of course, any ecologist at the time, is confronted with this rather tragic situation, which is, if you go to outer space, you see that the planet is filling up with streetlights. We've hit peak baby already. We're going to top out at the end of the century at 10.2 billion people, and of course with it there's not going to be room for much else. We're going to wipe out the biodiversity that the 2 billion years worth of natural selection has evolved, and I don't really see there's much around that. A very large chunk of our biodiversity is going to be gone, and I think the real tragedy is that the value of biodiversity is not in all the things that people talk about in the context of global climate change, in terms of, ecosystem services and the like. The value really in biodiversity is the fact that all of these different organisms, most of which we have not described, we don't know anything about their natural history, we have not even begun to sequence them, they have all solved some really interesting problem to live on every possible niche there is on this planet. And so there's all these incredible biological solutions out there that we're still clueless about, and that really Mother Nature is the mother invention. And it always has been for us. Most of what we think are human innovations are really copycat things from nature. And I don't need to go into examples. I'm sure that you know this probably for yourself. The problem is that we have trained a group of biologists who no longer know how to access that interesting part of our natural history legacy. Throughout the evolution of human beings, we have been naturalists. We have a nervous system that is extremely well designed to organize natural history information. Our brain just does that automatically. If we didn't, we would become lunch for something else. If we didn't, we wouldn't be able to eat lunch, right? That's been the nature of being Homo sapiens. We don't have any teeth, we don't have any claws, we have to have a brain to be able to outsmart things, to be able to eat them and be the heterotroph that we are. And then what happened when we domesticated plants and animals, we domesticated ourselves. And we lost that natural history sense completely. Most of the people who are heterotrophs on this planet don't know where their food comes from. And I'm afraid to say that's true for biologists too. As most biologists know the model system they work on, period. That's it. It used to be 20 years ago that you could take almost any biologist outside and they would know a large number of the species outdoors, but that's not true anymore. And so Tom Eisner and his nature vision really was the one who gave me this idea that you have to go forward, you got to go backwards. You have to go back to a time when we were training naturalists the way that Carl Linnaeus and Charles Darwin were trained, in a way to be able to understand how organisms work in nature, and to be able to, of course, combine that together with all the wonderful things that have happened in the toolboxes of biology. And chemical ecology is a discipline that brings together natural products chemistry and ecology. And what I wanted to do is to bring molecular biology into that so that you could manipulate the chemistry that the organism produces via the manipulations of molecular biology, and to merge them together. As I mentioned to the person who was interviewing me yesterday, that in order to make this happen, you have to break down disciplinary barriers. And those three disciplines are usually taught in universities in completely different departments. There's not a lot of cross-communication, and particularly amongst ecologists, for reasons that we can go into later. Ecologists have just not participated in the molecular biology revolution in any meaningful way. They've been in different departments, they speak a different language, and it's been very difficult to make this fusion of these disciplines together, working with mature postdoc scientists. So I've done most of my work with PhDs, because they're not yet got the blinders on, and created platforms that allow early stage scientists to be able to use all those different tools. And I call them a different species. I call them these genome-enabled field biologists. But the Catholic Church was right. If you want a conversion, you have to start early. If you start late, it's hard to make a real conversion. So it's been a real pleasure working with the young scientists, but the field has moved on, and I think now we have many labs doing this now. So what I want to do I've got 10 minutes, very rapidly give you just a bit of a flavor of some of the things that we do. We work on this particular native tobacco plant, which has this really interesting germination behavior that chases fires. And because it chases fires, it grows in the primarily agricultural niche. And by studying it, we hope to make crop plants smarter, more capable of being able to handle their own ecological challenges without having to be taken care of. So we have this field station in Utah. We do all this stuff that you can do so wonderfully in the German system, extremely precisely, extremely analytically, but we can't do any fieldwork GMs in Europe, so we take them to the US in a field station where this plant grows and is a native habitat. And thanks to the Brigham Young University, this is research that we probably couldn't do at any other American university. And we have all these field plots out there that we bring the transformed plants to plop them into these field setting that's right in the middle of their natural habitat. Here's all the different transformed lines we've been releasing over the years. Just to show you, we tried a little bit in Germany, but then it became outlawed to do that here around 2007. Frau Kunanz passed this law that made it almost impossible to do. So all of the work has been all the field work has been emphasized in the US. Sometimes we release into natural populations. APHIS regulates this, and we really enjoy being able to do it. But the basic philosophy is that we take all the organisms that interact with this one particular plant, and we use all of those interactions to help phenotype the genes in this particular plant. So we use nature as a laboratory to study gene function. Now, genes are either maintained or pseudogenized from a genome based on their effect on the plant organism's Darwinian fitness. And you frequently read in the literature, oh, here's another example of gene redundancy. Well, I would argue that I would bet that a large fraction of the redundant genes that we call genes of redundant function is simply just the reflection of how shallow we phenotype these plants, or the organisms, whatever organism you're dealing with. Almost all of the genes that we have silenced and put out into the plants have no phenotype at all in the glasshouse, but they have beautiful phenotypes when we take them out into the real world. And I think this is where, if you want to understand gene function, you have to be doing manipulations in the world where the organism evolved. So we use nature as a laboratory, and we're using basically all of these different organisms to tell us different things about this plant's genome. And we've got lots of examples of bacteria and various types of insects that have identified the functions of genes and told us different mutants of natural ones. And I could go on for hours and tell you stories about this. And I really just want to get to one very important point about this process, because it gets back to the business about being science, about being a knowledge addict. To maintain a knowledge addiction for a lifetime requires that you keep somehow that dopamine loop activated. And what typically happens in the development of a young scientist is that you grow a big lab, and before you know it, you haven't been in the laboratory for weeks, months, and then years go by. And then you start getting more and more separated from the actual excitement of the experimental process. And I think that is an extremely dangerous thing if you want to be a scientist for the long haul. It's just not possible to have the same level of enthusiasm when you're just trying to manage people or edit manuscripts or go to congresses and talk about other stuff. There is nothing that substitutes for the primacy of being able to be part of that discovery process, have your own hands, your own mind, be involved in it. For me, the way I do it is that 4 months out of every year, I go to the field station. I keep track of all those natural history observations. The natural history stuff is by head. Natural history does not get old. It always is like a good wine. It just gets better with age. Every year is a different discovery, and it brings in, new perspectives on phenotypes of the plants that we do. And I think the key thing to becoming and maintaining a vigorous scientific profile for a career, and this is particularly true if you work in a research institute where you have no other job you could go to. In universities, at least, if you get burned out on research, you could become an administrator, a teacher, a mentor, or lots of other sorts of roles you could do. But in a research institute, if you're not hot on research, you got to fake it. And I can't tell you how many, Max Planck directors I see are faking it because they lost the burn. They lost that connection. So I guess if I was going to give a piece of advice, think about some part of the research program that you have that you can still and maintain direct connection to, have your hands on. Ron mentioned it in his talk, and I think in the talk we heard from, Fred, thank you. I'm having a verbal block here. Fred gave a beautiful example of how he had kept track of the quantitative microscopy he had done throughout his entire career. And I think having some aspect where you are in direct contact with the technology, and the technologies which is constantly going to be evolving, is such an essential part of being an active scientist. As I say, for me, natural history has been the thing that I keep track of. I do a lot of mass spectrometry. I went from automobile repair to mass spec repair, so mass spectrometry was a major part of my group building activity. And since I only have 5 more minutes, I think instead of going into a lot of research, which I had hoped to be able to do and highlight all those wonderful Indian students' results, I think I'll probably just end it here and open it up if there's a question or two we can do. Is that all right? Thank you for your attention.